Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Assay for Transposase-Accessible Chromatin using sequencing, a comprehensive guide to chromatin accessibility and single-cell analysis. Table of Contents. Introduction to Assay for Transposase-Accessible Chromatin using sequencing. History, Principle, Steps, Advantages, Limitations, Applications. Single-cell assay for transposase-accessible chromatin using sequencing. Introduction to Assay for Transposase-Accessible Chromatin using sequencing. Assay for transposase-accessible chromatin using sequencing is a method used to study chromatin accessibility and gene regulation. This method identifies open chromatin regions where regulatory proteins can bind. It provides information about how DNA is packaged within the nucleus. A ATAC-Seq helps us understand how chromatin structure can influence gene expression. DNA is organized into three hierarchical structures: nucleosomes, chromatin, and chromosomes. Chromatin exists in two dynamic states and can transition between transcriptionally active euchromatin, open, and inactive heterochromatin, closed. Open chromatin regions are usually linked to active genes and transcription factor binding. Studying chromatin structure is important because it plays key roles in gene regulation. Different sequencing methods have been developed to study chromatin accessibility and epigenetic modifications. These methods include DNase-I hypersensitive sites sequencing, formaldehyde-assisted isolation of regulatory elements sequencing, chromatin immunoprecipitation sequencing, and micrococcal nuclease sequencing. Among these methods, ATAC-Seq has become widely used in epigenomics due to its efficiency and minimal input requirements compared to traditional methods. ATAC-Seq has a simple workflow. It requires a small number of cells. It provides detailed information about regulatory elements. Since its introduction, ATAC-Seq has been used in different biological fields like disease research, epigenetics, and cancer studies. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. History of ATAC-Seq. A TAC-Seq was introduced in 2013 by Jason Buenrostro and his team at Stanford University. Earlier, epigenetic methods like DNase-I hypersensitive sites, sequencing, and formaldehyde-assisted isolation of regulatory elements. Sequencing required large amounts of starting material. These earlier methods had complex protocols. They could not analyze all three chromatin features simultaneously in a single assay. Chromatin immunoprecipitation sequencing also had limitations, as it required prior knowledge of the specific epigenetic mechanisms involved. To overcome these limitations, ATAC-Seq was developed as a simple and efficient method. ATAC-Seq requires far fewer cells compared to previous methods. It provides a genome-wide view of chromatin accessibility. This method has improved over time, leading to innovations such as single-cell ATAC-Seq. Single-cell ATAC-Seq helps study chromatin accessibility at the level of individual cells. Recent improvements combine ATAC-Seq with other techniques like RNA sequencing and DNA methylation analysis. These combinations provide more detailed information about gene regulation. Principle of ATAC-Seq. ATAC-Seq works by using an enzyme called TN5-transposase that simultaneously cuts DNA and inserts sequencing adapters. This combined process is known as tagmentation or transposition. Tagmentation targets open chromatin regions. The T.N.5 enzyme selectively inserts into accessible DNA. Sequencing the resulting fragments identifies open chromatin regions. These open regions are where genes can be accessed and regulated. ATAC-Seq has three main steps before sequencing: cell lysis, transposition, and amplification. In the first step, cell lysis isolates nuclei from the sample and prepares them for transposition. During transposition, the TN5 enzyme cuts open chromatin regions and inserts sequencing adapters into these accessible regions. The tagged DNA fragments are then amplified. These fragments are prepared for high-throughput sequencing. The sequencing data is analyzed to identify regions of the genome that are open and accessible. Steps of a TAC-Seq. Cell preparation. The first step in ATAC-Seq is isolating and preparing cells for analysis. In this step, cells are harvested, counted, and lysed to obtain pure nuclei. It is important to maintain single-cell suspensions for accurate results. The cells are lysed using a lysis buffer. The lysis buffer breaks open the cells and releases their nuclei while keeping chromatin intact. After lysis, the cells are centrifuged. The cells are then washed to remove excess buffers. Transposition. The obtained chromatin is fragmented and tagged with sequencing adapters using the T.N.5 transposase enzyme to create the ATAC-Seq library. TN5 cleaves the chromatin into smaller fragments. This process specifically targets open chromatin regions. After fragmentation and tagging, the library is purified. Purification removes excess transposase and contaminants. Amplification. The purified DNA fragments undergo PCR amplification to increase the quantity of DNA for sequencing. PCR amplification ensures there is enough DNA for high-throughput sequencing. The final DNA library can be checked for quality. Quality control methods include gel electrophoresis or fluorescence-based detection methods. Sequencing. The final ATAC-Seq libraries are loaded into a sequencing machine. The most commonly used sequencing platform for ATAC-Seq is Illumina. Paired-end sequencing is often preferred because it sequences both ends of the DNA fragments. Paired-end sequencing provides more information about fragment size and the location of accessible regions. Single-end sequencing can also be used. However, single-end sequencing provides less structural information and is less accurate. Data analysis. A TAC-Seq data analysis consists of four main steps: pre-analysis, core analysis, advanced analysis, and multi-omics integration. Pre-analysis involves quality control and sequence alignment. Pre-alignment QC checks the sequencing data for errors or unwanted adapter sequences. Tools like FastQC are used to check base quality, GC content, and repeated sequences. Trim-o-matic is used to remove low-quality reads and adapter sequences. After trimming, the cleaned reads are aligned to the reference genome. Post-alignment QC filters out low-quality reads, removes duplicates, and checks fragment sizes. Core analysis includes peak calling, which identifies open chromatin regions. Most peak callers for ATAC sequence are adapted from chromatin immunoprecipitation sequencing or DNase I hypersensitive sites sequencing methods. Hidden Markov Model R for ATAC-Seq is the only peak caller specifically developed for ATAC-Seq. Advanced analysis identifies 4 key patterns in the data: peak, motif, footprint, and nucleosome. It includes annotating peaks, detecting transcription factor motifs, identifying transcription factor footprints, and studying nucleosome positioning. ATAC-Seq data can also be combined with other omics datasets to fully understand gene regulation. Multi-omics integration enhances understanding by comparing ATAC-Seq data with chromatin immunoprecipitation sequencing to confirm binding sites. It uses RNA-seq to link chromatin accessibility with gene expression. It also aids in reconstructing regulatory networks. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Advantages of ATAC-Seq. A TAC-Seq requires fewer cells, making it ideal for studying complex or rare samples. It has a simpler library preparation method, which makes it easier and faster to perform. It does not need prior knowledge of epigenetic mechanisms, unlike earlier methods. It has high sensitivity and can accurately detect open chromatin and regulatory elements at both the bulk and single-cell levels. It provides fast results, making it suitable for large-scale applications like clinical research and drug development. It can be used to understand diseases linked to epigenetic changes, including cancer, autoimmune diseases, and neurodegenerative disorders. Limitations of ATAC-Seq. A TAC-Seq only captures chromatin accessibility and does not provide information about DNA methylation, histone modifications, and chromosome interactions. The TN5 transposase used in ATAC-Seq can introduce bias in adapter ligation. TN5 can sometimes insert the same adapter into both ends of a DNA fragment, resulting in unusable fragments for sequencing. This can lead to data loss and uneven coverage of the genome. The PCR amplification step can create challenges when dealing with large DNA fragments. A TAC-Seq faces computational challenges as only a few bioinformatics tools have been developed specifically for ATAC-Seq data. Researchers often rely on tools designed for chromatin immunoprecipitation sequencing and DNase I hypersensitive sites sequencing for data analysis. Applications of ATAC-Seq. A TAC-Seq is primarily used to map open chromatin regions, enabling the study of genomic regions accessible for transcription. These maps help identify regulatory elements such as enhancers and promoters that play crucial roles in gene expression. It is extensively used to investigate gene regulation and transcription factor binding in various biological processes, including complex diseases, immune responses, and cancer. A TAC-Seq is also employed in nucleosome mapping to understand how chromatin structure influences gene activity. It has applications in cancer epigenetics, where it is used to explore chromatin accessibility changes in cancer cells. In disease research, ATAC-Seq helps map disease-specific chromatin alterations, identify regulatory elements linked to diseases, and uncover potential therapies targeting chromatin accessibility. The technique is also used in comparative epigenomics and evolutionary research. Single-cell ATAC-Seq. Single-cell ATAC sequence is an advanced variant of ATAC sequence that enables a study of chromatin accessibility at the level of individual cells. Unlike traditional ATAC sequence, which analyzes bulk cell populations, single-cell ATAC sequence provides insights into cell-to-cell differences in chromatin structure. It allows for the identification of chromatin accessibility in thousands of individual cells using a simple and cost-effective protocol. Single-cell ATAC sequence utilizes techniques such as fluorescence-activated cell sorting, microfluidics, and nanowell platforms to isolate and analyze individual cells. After isolating the cells, the ATAC sequence process is performed on each cell separately. By focusing on individual cells, single-cell ATAC-Seq uncovers details that may be obscured in bulk ATAC-Seq data, making it valuable for identifying rare or hidden cell types. It also helps in understanding how different cell types are regulated. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!